It's for me really a great pleasure and an honor to receive today Dr. Leon Chu, Director of the Advanced Imaging Center at the Howard Hughes Medical Institute in Janelia Research Campus. For the last 10 years or more, Leon has been impulsing and promoting programs at this place who are specifically targeted to people outside Janelia, for people who is all over the world, to take advantage of the truly cutting-edge technology that they develop at this place. And in particular, I want to highlight some, like the optical imaging across bioscale length workshop, who have first edition this year, and we all hope that we will have further edition the next years. But also Imaging Africa and Imaging Latin America, and it's the reason for why all the students are here, and Leon and all the crew from Janelia came down to Uruguay, Montevideo, to kick off with this first meeting for all Latin Americans. Leon has promoted the broadening of the outreach of Janelia over these years, and he will tell more. I hope this will be even broader and broader over the time. And he also launched a particular fellowship that is targeted to people after PhD, fellowship for 2 years, and get trained not only in imaging but also in how to deal with core facilities, etc. Leon serves as a scientific advisory board of many bioimaging initiatives. In particular, I want to highlight Euro Bioimaging, France Bioimaging, and Africa Bioimaging Consortium, and is currently the chair of the Global Bioimaging Manager Board. The Janelia period, Leon, 17 years at the Advanced Microscopy Center in Northwestern University, and through his career, he has specifically interest in the understanding of the dynamics and organization of cytoskeleton in terms of biology, with a lot of focus on imaging. At the end, I want to mention basically a couple of things who are really, in my opinion, and I want to highlight. From a person who is in one of the places that if you want to do imaging, this is the place, he has spent a lot of energy and effort in impulsing and promoting imaging in regions where there is not such opportunities like the one we or other people may have in other places. And I want to say he's the founder of Imaging Africa Workshop, and he's also the person behind the Africa Microscopy Initiative, who is not only an initiative to bring to Africa imaging technology, but also to build capacities, human capacities, that can promote and disseminate all the possibilities that imaging offer for science. Leonie will tell more about this. This is an amazing initiative, and for me, a pleasure and an honor that since the last week, Leon was here, Karen Sheiko from Africa was here, Karen Sheiko with Microscopy at Leon Lab. They are launching this imaging center and African Microscopy Initiative in Cape Town, and they will be cutting the ribbon next October. And we shake hands between Karen and me to put together the Advanced Imaging Center in Janelia, the imaging core facility in Africa Microscopy Initiative, and the Advanced Bioimaging Unit here in Uruguay, to be sister core facilities and try to use this friendship and this community to promote all the opportunities between these three centers. So thank you very much, Leon. It's really a pleasure for me to have you here. I pass the word to you.
and that is where all the imaging happens. From the foundation up, Uruguay has played a huge part in this building. It's designed by Gabriel Vignoli. So this is the artwork almost for science, a tribute to science by a fellow Uruguayan. So what I'm going to talk to you about is the Advanced Imaging Center at Janelia. And what makes this imaging center unique is that nobody at Janelia is allowed to use it. This is purely for you. So you have to consider this as a technological extension of your local imaging center. You push your biological questions as hard as you can using whatever technology that you have. If you really have that need to go one step further, that is when you write a proposal and come to Janelia. And I will definitely discuss the program a little bit. So this is both a toy talk, technology talk, as well as an opportunity talk. So as I go through the technology, try not to listen passively, but start thinking about how this technology can be used in the experiment and the questions that you are trying to address at the time. So let's start with something we all know very well, which is the GFP molecule. Everybody has used GFP to tag something, and then you're trying to follow how it interacts with one another and try to understand the dynamics of the protein. But the one problem with any regular conventional microscope is that this is a light coming from a single GFP molecule. But if you actually shrink that molecule down to its actual size, or as small as PowerPoint allowed me to shrink it, that is the size of a GFP molecule. And that is the light that GFP gives you through a conventional, even with the high resolution microscope. So it calls into questions a lot of the colocalization studies that you see in literature, but more importantly, you can imagine you can fit a lot of GFP molecule into just that point source. So this is a huge problem, obviously, and this is when in 1994 that Eric Batzig came out with this thought experiment, single author paper. No experiments whatsoever. Isn't that nice? So he basically thought, well, if you have a conventional level of resolution, and this is the best a microtubule would look like under a regular microscope, but if we can actually activate those one molecule at a time, and then resolve it, Gaussian deblurring it, into the right position, you can localize a lot of molecule, and all you have to do is to do it 20, 30,000 times, right? So this is nothing new in 2014. This is one of the techniques that won the Nobel Prize for super resolution development. Now, while you can resolve this into 10, 20 nanometer resolution, this technique only allows you to do this on a 2D plane. And this is equivalent to you putting dots on paper. The inconvenient truth is that light happens to happen in 3D. So that's very unsatisfying to do resolution like this. And so we have to add resolution, preferably isotropic resolution, also in the axial plane. So the X, Y, and Z resolution is the same. And if you think this is hard, adding resolution in the third plane is actually significantly harder. And usually when microscopists run out of idea, we tend to turn to astronomy because they are light years ahead of us, all pun intended. So if you are an astronomer and if you want to quantify the distance of this quasar that is pulsing that consistent radioactive radio wave, what you will do is you put two radio telescopes on each end of the planet. And after watching this, you should never complain that a microscopy experiment is difficult. And so by detecting almost that delta T that these two radio telescopes detect from the same signal, you can actually put those two signals together and let those signals interfere with one another. And the degree of interference will tell you the distance. And that, is the nutshell of that math. And that mathematical manipulation is called interferometry. And so what Harold Hess did at Janelia is they basically shrink that millions of light years of distance into a microscope and created this monster. And this is the interferometry palm system. And as complicated as this microscope look, from a wiring perspective, the principle behind it is actually elegantly simple. So this is actually the sort of the light path of the IPON. I'm going to walk you through it. It's actually not as complicated as it appears. So this microscope has two objective lenses sitting in opposing position, and your sample will be sandwiched in between the two cover slips. And then when the sample is excited, the emitted fluorescent light will be channeled into this diamond-shaped beam splitter. It will be channeled by the lower objective, and it will also be channeled by the upper objective into this beam splitter. This is where the magic happens. And this is the enlarged picture of that same three-way beam splitter. And I'm going to show you how it works. So you actually have three horizontal lines. So the first horizontal line is a 6633 beam splitter. What that means is that when the light hit it, 66% of the light will go through, 33% of the light will be reflected. The second horizontal line is a 50-50 beam splitter. Half of the light will be reflected, half of the light will be transmitted. The last one is a mirror, all the light will be reflected. And if you do a simple calculation at the back of an envelope, you'll very quickly come to the conclusion that exactly 1/3 of the light will come out and hit CCD1, the other third will come out and hit CCD2, the last third will come out and hit CCD3 camera. So if your molecule is dead center in between the two objective lenses, the light will hit CCD1, 2, and 3 simultaneously. So there will be no phase change. And when you put those signals together, they will not interfere with one another. But as soon as that molecule is off-centered, even by a little bit, you put those signals together, they start to interfere. The more off-centered, the more interference it's going to create. And from that point, we just brute force calculate exactly where that molecule is in the Z-plane. So this actually creates a super resolution microscope where the Z resolution is even better than the XY resolution. The XY resolution remained at about 20 nanometer or so. The Z resolution is now about 10 to 12 nanometer. So what can you do with this kind of microscope? So here's an example. This is from Johanna Ivaska in Finland. And most of you have used confocal before. You take a confocal, you can section a single cell into multiple slices. What we're going to do here is we're going to slice a single protein complex into multiple slices. And in this case, you're looking at 5 focal adhesion. The red is paxillin, and the green is one of the signaling molecule called KENK1. And so you are actually going to see optical sectioning going in and out, in and out, in and out of that 5 focal adhesion at 10 nanometer or so step size. And we're going to tilt it to the side. You actually now get to see the height of the focal adhesion. Paxillin being very close to the extracellular matrix, the signaling molecule KENK1 in green is much higher. For the first time, we are able to resolve completely using fluorescent microscopy the inner and the outer membrane of a single bacterium. And the outer membrane is red, the inner membrane is green. So this is the kind of super resolution that we are able to do with the interferometry palm system. Now, every time you image at this kind of resolution, you pay a high cost at it. There's no free lunch in microscopy. You always sacrifice something. And in this case, the one factor that you absolutely have to achieve is the labeling density, because when you're imaging at something that deep, any space that is not labeled is going to be very jarring, and you may not be able to join all the information together to create an ultra structure of a biological target of interest. And in fact, the density of labeling that you need is about 100,000 molecule in a diffraction limited radius space if you are going to image at 10 nanometer resolution in 3D. So that is very, very high density that most biologists will not be able to deliver. It's just not possible. So that will always be a limitation here. So that sort of leads us to this tetrahedron of frustration, right? And so every time you gain something, you always pay a price and sacrifice something else. And in this case, IPOM achieved an extremely high spatial resolution, and it immediately pulled itself away from the imaging depth. IPOM is a turf-based microscope, so it only have certain penetration depth. It takes about an hour or so to take an image, so there is no temporal resolution to talk about. I mean, the sample is fixed anyway, so tempo doesn't matter. And sometimes photo stability is also an issue. Another super resolution microscope developed by Stefan Hell and Max Planck, it's called STAD. When it was invented, the spatial resolution is somewhere around here, and by sacrificing the spatial resolution, STAD gained a lot of imaging depth, reasonable temporal resolution, but it also paid a huge price in photo stability. And in fact, the STAD microscope, by using a donut-shaped depletion laser to basically deplete all your emission coming from that point source until the donut hole is so small that the only light that can come through is through that donut hole. And so that is how that super resolution is achieved in Stefan Hell's system. And to achieve that kind of super resolution, you actually have to pump in about 10 million watt per centimeter square of power to your cell. And I don't want that 10 million watt per centimeter square of power to just go over your head. I actually want to give you an impression of how powerful 10 million watt per centimeter square actually is. So let's say if today you go to Sahara Desert and one of the hottest day possible, it is so hot that you feel like you're about to die. The sun is delivering to you at most 1 watt per centimeter square of power. You're expecting a naked cell to take 10 million sun under the microscope. And so you can't blame that the cell just choose to die. And your super resolution localization, the pointillism super resolution localization technique doesn't fare any better. We're still dealing with 10,000 sun, and if you think that your confocal is gentle, your confocal is somewhere around here. So it doesn't matter whether whether you're charbroil or you're grilled, you're still dead. So that is not good. And one of the, ironically, the most gentle of all the super resolution technique to date is the structural illumination microscope, and that did not win a Nobel Prize. So I'm gonna sort of, in this mad rush in the past 20 years or so, especially starting in the early 2000s, that everybody was chasing after higher and higher and higher super resolution. A lot of people lost sight of the fact that just by adding contrast to microscope, you will allow biologists to see a lot of details. It's not super resolution, but you see a lot of details. And in the past week, we have the class here, the workshop has covered the phase contrast and DIC. And in 1953, phase contrast technique got a Nobel Prize because simply that is how important adding contrast to microscopy actually is. So, biologist really doesn't care what is the smallest nanometer you can see. You care whether or not the biological stories that you are following can be addressed by the microscope. And so one way for you to achieve very high contrast in a fluorescent microscope is, instead of just excite the entire column of sample with a laser, how about we shape the laser into the form of a sheet and just excite only that plane that you are interested in imaging. And that gives rise to light sheet microscopy. So if you have a laser that is shaped in the form of a sheet, you just have to sweep that sheet up and down. And basically, since there is no other out of focus light being generated, so all the light can be collected by the objective lens and go straight to your detector without having to rely on a confocal pinhole, which will further reject informative photons, right? So again, this is nothing new. There are a lot of light sheet out there, so I'm going to show you a few things, and then I'm going to show you what we have available at Janelia that makes some of our light sheet extremely powerful. So this is actually one of the systems that we have, and what you are looking at here is a zebrafish embryo that has the neuron all labeled with one of the GCAM6 calcium sensor that is also developed at Janelia. And so what you're going to be seeing here is basically the neurons firing inside the zebrafish. So in a sense, we're looking at this zebrafish embryo thinking. I have no idea what it is thinking about. But at some point, you're actually going to see a burst of activity. So obviously, you got a little excited, probably dreaming about food. There you go. So this is some of the things that we can do. with the light sheet system. And you can also track something that is slightly more opaque, and in this case, the development of Drosophila embryo. So we can now track every single cell, the movement of every single cell in the developing Drosophila embryo. Okay. Now, this kind of microscope is now available widely. But if you are studying something bigger, for example, like a mouse embryo development, you're out of luck. Because when mammalian system develop, you start to form bone, you start to form skin, blood starts to flow, body cavity starts to flow. It's going to completely mess up your photon, because now it goes through multiple layers of different refractive index. You have no idea where that photon is coming from. So if you want to solve that problem, you have to create a smart microscope, a smart light sheet system, where it knows exactly how to adjust the angle and adjust the laser power and adjust the detector gain and everything, and even the position of the stage as that mouse embryo is rolling around, and to continue to track that without human intervention. So the microscope make the entire decision. And so to do that, we basically have created that system that has constant feedback to the light sheet system, so that you can continue to support that very long-term imaging. And so this is the system. Again, you have 4 objective lens. Those are the 2 excitation lens, and then these are the 2 detection lens, and your mouse embryo is sitting inside in between those 4 lenses. So the next movie I will show you is the very first time someone actually get to track every single cell in the developing mouse embryo. Here we go. Now, as you are appreciating this movie, I also want you to pay attention to the right side. And as the mouse embryo get bigger, you start to see all the correction kicking in. So the microscope is making its own decision and continue to track that mouse. If we turn off all this autopilot system, this mouse embryo is out the door. It's rolling so far away. It's not only out of the screen, it's out of the door. So we'll be able to keep this kind of sample continue to develop. And here is another example. So all the pink cells that you see here are endothelial cells. And at some point, the endothelial cells will differentiate and become cardiomyocyte. And when it becomes cardiomyocyte, we flux out one of the gene and it turns green. So you're actually looking at differentiation of endothelial cells into cardiomyocyte, and at the same time, you're going to also see the cardiomyocyte form a heart. And so it's actually easier to look at the gray area. So you start to see this heart tube starts to form. First set of chamber, second set of chamber, and the heart starting to beat. And so now we can also track the direction, the velocity of every single cell in 3D, in this really kind of beautiful and mesmerizing choreography of all the cells. And you realize that the cells don't always move in homogeneous direction. Sometimes you have a burst of extremely high speed in certain area, and then they cool down, and then some other cluster takes over as that mouse continue to develop. All right, and this movie goes on forever and ever. So this is all nice and good if you are a developmental biologist. If you are a cell biologist, you are also out of luck, because most of the light sheet that I've described up to now, and a lot of the light sheet out there, uses something what we will call a Gaussian beam. So as the name suggests, the Gaussian beam is actually pretty fat, because it actually adheres to the Gaussian profile, as in the red Gaussian bell curve. So as it propagates deeper and deeper and deeper into the specimen, it actually gets thicker and thicker and thicker. So it is so thick that sometimes it could be microns thick. And if you're a cell biologist, that the entire light sheet is sometimes thicker than your cell, then it has no sectioning capability. You don't want that. So we have to create a way where we can make that light sheet thin enough that you can actually use it for a single cell. And so we can no longer rely on what we'll call the Gaussian beam. We have to change to a completely different kind of laser beam called the Bessel beam, which adhere to the Bessel mathematical profile that has this very long, thin pencil, thin line profile. And so just to show you what we talk about, that's if you're using the conventional Gaussian beam, if you are using a very good high NA lens, you will get a pretty thin laser sheet, but it doesn't propagate very far. If you want it to propagate very far, it starts to get thicker and thicker, and then you lose resolution. Whereas with a bezel beam, you can continue to keep it very long and very thin at the same time. Another really cool features of bezel beam is that bezel beam looks like a bullseye. It has the center point, it has a lot of concentric circle. If you pack more and more and more and more bezel beam together, guess what happened? They start to interfere and create all this crazy pattern, and at various points, you actually see the creation of light sheet. You just missed one. Here's another one coming. Here's a light sheet. It just disappeared. And then another one coming soon. Here you go. So by packing a lot of bezel beams together and let those concentric circles interfere with one another, you start to create a very, very thin light sheet. And so in that sense, what we are doing here is to create a lattice light sheet with a lot of bezel beam packed together. Now we truly have a light sheet microscope that is designed for a single cell. And so what kind of movie can you see? So the very first that what I'm going to show you is, so this is that lattice light sheet microscope at the Advanced Imaging Center. And then what you're looking at here is a T cell in orange interacting with an antigen presenting cells. So this is actually also the very first time people actually see an antigen presenting cells and a T cell interacting with one another. And you also see a lot of this finger-like protrusion where the two cells interact, where the T cells sends out a lot of those protrusions. Those are immunological synapses. All right, so obviously this is one heck of an angry T cell. Here is another example by Lakshmi from NIH. Same, similar situation. But here we're staining a component of a T cell receptor and a signaling molecule in two different colors. So when I play the movie, you're going to see this T cell come up and touch the antigen presenting cell. But before they touch, you do not see any yellow. Yellow actually signifies that the T cell receptor complex has interacted with the very last signaling component cascade of a T cell receptor activation cascade. So when you see yellow, T cell receptor has been fully activated. Here we go. No yellow, touches. A few seconds later, yellow shows up. T cell receptor activation complete. This is the exact same event. Now I invite all of you to imagine that you are the antigen presenting cells. The T cells come and form T cell receptor complex in your face. So yellow, all the immunological synapses come in. And here is another example. Now with lattice light sheet, because it is so thin that you actually, when you excite only a very thin layer of biological material, guess what you get? You get an extremely high contrast, because not a lot of things are, messing up your perception. And so you actually get to see a lot of biological details that you usually don't get to see. And in this case, this cell is about to divide. This is by Erika Holzbauer, last year's president of ASCB. And this, you actually start to see, as this cell is preparing itself to divide, you also see this haze of blue, hopefully you guys can see, that's actually actin, a cloud of actin that nobody has ever seen before, as if it is shuffling the mitochondria so that when the two cells divide, that both cells get almost equal amount of mitochondria in the two daughter cells as the thing is dividing. And also in collaboration with Klaus Hahn at UNC Chapel Hill, we are starting to combine FRET imaging with lattice light sheet. And the top, we're looking at the GAF-H1 activity in mouse embryonic fibroblast. And we are looking at how the GAF-H1 activity, when it is activated, it turns red. And you can see that GAF-H1 is quite activated in the membrane ruffle. And then the bottom cells, they are interested in looking at RAT1 activity, but in this case, Klaus is not interested in the surface. He wants to look at it in the cytoplasm. So we basically peel away the cytoplasm to show you where the RAT1 activity actually is located inside, under the hood. And we can also take it up a notch in the biological scale of complexity. We are looking at dorsal closure at the back of a Drosophila embryo. And so we're going to play through this movie, and the cell is stained with cadherin, I believe, yes. And then the abdomen is starting to form into a 2-column, and this is how the lattice light sheet scans through the sample, slow down almost 1,000 times. Now, as you are watching the choreography of this biological system, I also want you to pay attention to the number of Z-stack that we are collecting. Usually, on a confocal microscope, you're pretty happy if you get 20, 30 stacks. This goes up to 800. The record at the AIC is 25,000. I always like to joke, but unfortunately it's not a joke. It was an experiment stopped by a very angry phone call from the IT department. We generated way too much data. But so the fact that this system can go on and on and on and on is precisely because it is extremely gentle. And the reason why it is gentle is, again, because the light sheet is very thin. And when it is thin, you have a lot of contrast. When you have a lot of contrast, you see details. When you see details, you back off from the laser. So at the end, this is probably one of the most gentle illumination instruments out there. In fact, for those of you who are cell biologists, you know that one of the most important acid tests for phototoxicity is whether or not the cell undergoes cell division. Any cells that has phototoxicity will be arrested at the cytokinesis phase. They will never divide. Well, we'll see. And it just keeps going on and on and on and on and on. And it, in fact, go on so much that I cannot believe I actually say that. Like, this is getting a little boring. And that is done by Edouard Bertrand, who is the president of the current president of the France Bioimaging. And this is just the preliminary data. What he really wanted to do is captured by this movie. So he's really interested to simultaneously coordinate the visualization of gene transcription as well as cellular behavior in the whole organism. So what we are doing here is, you're going to see those things divide, and then you also start to see the appearance of pink dots. The pink dots are actually loci of the genome that is marked by the MS2 probes, and those loci actually encode a protein that will affect cellular behavior that will induce dorsal closure. So now you actually see gene expression, protein expression, then followed by cellular behavior, all in one single movie, from molecular biology all the way to developmental biology in one shot. So this is all nice and good in Drosophila, but sometimes we want to go deeper and go into larger organism. Now, as you go deeper and deeper into larger organism, you're going to run into all sorts of problems. So this is actually a 2-photon scan of a brain. The two common problems that you will encounter is basically light aberration and scattering. The tissue is just going to mess it up. So to solve this problem, we once again turn to astronomy. If you are an astronomer, you have an earthbound telescope. You look into the sky. The light coming from the skies will have to go through all the distortion of the Earth atmosphere, and then it's going to get distorted, right? And twinkle, twinkle, little star is just a romantic way of saying light aberration, just so that we don't scare the hell out of kids. So to solve this problem, and the astronomers basically have to fire a very powerful laser about 90 kilometers up into the sky, where the laser sees the sodium layer. And when sodium sees laser, it fluoresces. So what the astronomers are trying to do is to create a guide star that is actually not there. But the light coming from the guide star will have to fight through the same atmospheric distortion, so the wavefront is completely distorted. But since you are the one who point the laser up there, you know exactly where that point should be. So all you have to do is to create a counter wave, same amplitude, different direction, using a deformable mirror under which you have many mini pistons. So you create a counter wave. So by creating a counter wave, you can straighten out all the aberrated wavefront. Now you can do Earth-based telescope observation without any aberration. So that technique is adaptive optics, and that is what we are going to put into the microscope. All right. So this is a 2-photon image of a Drosophila brain, actually a zebrafish brain. conventional two-photon microscope, but the data is now corrected with the adaptive optics. So we're actually going to rebuild the entire brain of the zebrafish. And the neurons here are stochastically labeled, so there is no specific lineage here. So here's the entire brain of the zebrafish. And now we are actually going to do a flyby. So we're going to go into the brain. And what we're going to do when we're inside the brain, we are going to turn off the adaptive optics and show you what the reality actually is. The reality is actually pretty bad. That is the reality. But with adaptive optics, you correct it all, okay? And also with that, it actually highlights how bad sometimes microscope can distort reality. And you always hear that, you know, seeing is believing all the time. So you have to start thinking whether can you believe anything you see. So this is a notochord of a zebrafish. And as I peel back the notochord, you see all this potato chips looking thing. These are the distorted wavefront of your laser as it is going in. Even your laser is distorted as it is going in. So this is some pretty serious distortion. And then we're going to pull it back and then pull it back one more time. Now what you're seeing is the detection wavefront. So both the emission and the excitation wavefront is completely distorted. So this is what imaging is usually done. And so you are looking at a lot of distorted images. But now that we have all this data, we can now do correction, right? And so this is the instruments that we built at Janelia. So it actually has quite a lot of modality, which I'm not going to go into, but the most important modality here is the lattice light sheet with the adaptive optics. And I'm going to show you a rendition of it so that you can actually see the various parts. This is our attempt to try to create a human. So this is your objective lens, just like a lattice light sheet lens with an excitation and a detection. At the bottom there is another detection objective. Going over there is a two-photon objective, which you can also combine with adaptive optics to do two-photon. And all the way up there, and the third floor is the detection light path, followed by all the emission, excitation, filtering light path, and as well as all the filtering positions. And at the bottom is the housing for the laser. You can clearly see the two-photon laser here, which we use to generate that guide star inside the tissue. So let me show you one of the movies. So this movie actually made it to National Geographic magazine. So this is a lymphatic blood vessels between the ear and the brain of a zebrafish. So this is a lymphatic space. And this is before adaptive optic correction. This is after correction. Now you see the entire endothelial wall. There's also some junk that macrophages like to eat. Immunosurveillance. And I'll let you count how many times macrophages go through the endothelial wall as if the endothelium is not there. And if you're trying to do this on a plastic dish, you're praying and praying and hope that the macrophage go through your endothelial cells, but in real life, they just go through as if it's not there. And this is actually a very important lesson, is when you finally engineered an instrument where the illumination power is at the level similar to that under which life evolved. The beauty and the magic and everything of life comes to you, and you actually get to see exactly how life processes played out. And I think with this kind of microscope, it also now ushered in an era, a very, very important era, where the 100-year-old dream of cell biologists to be able to perform cell biological experiment in the context of a whole organism, I think, has arrived. And here is a very complicated chunk of tissue at the forehead of the zebrafish, and it's just a messy chunk of tissue. So we are going to pull all the cells apart digitally. So this certainly takes segmentation to a different level. And you can actually see dividing cells inside the zebrafish. And then what we're going to do is to focus on a single cell, and hopefully I will be able to prove to you that we can track 3 or 4 different organelles inside a living cell, inside a living organism, simultaneously. And so you start to look at mitotracker, you're looking at Golgi, you're looking at ER simultaneously. Actually, at some point, that cell is going to divide as we are tracking it inside a live animal. In the next cycle, you see it is going to divide. It is about to divide. Boom, it divides. Right, so this is the lattice light sheet with the adaptive optics. So the last instrument I'm going to introduce to you is now we're going to go to the dark side of imaging where no photon is involved, EM. So in the good old days, if you want to do volumetric EM, you have to use a microtome. If people who are very good with their hand can basically generate a beautiful ribbon, and you hope to God that you don't sneeze during the next half an hour and you're trying to bring those things back. But there are now various ways for you to mill and image. There are two ways to do that. One is using a serial block phase that is much faster but has lower resolution. And another one is using focused ion beam SEM. Now, focused ion beam SEM works by you using, a focused ion beam to mill away a very, very thin layer of material, and then there's an SEM there, take an image, and then mill another layer, take an image. Like a confocal, it's just that it physically removes stuff. Commercially, there are 3 or 4 focused ion beam SEM out there, but the most they can continuously image is about 3 or 4 days. And what we want to be able to do at Janelia is to create a focused ion beam SEM that can mill until the entire filament run out. So we're talking about continuous, non-stop 24-hour imaging and milling for months and months and months on end. And so Harold has successfully generated that. And here is an example. So this is actually a very short run. This is only about 2 or 3 weeks or so of run. So this is actually a clammy. And when I play through this movie, you can watch this area where you will see the cilia coming out. You will also see the 9+2 structure being cut longitudinally. There we go. Here comes the 9+2 structure. All right. And then here's another sister 9+2 structure over there. And then now we are milling through the entire cell. And as we're going through the nucleus, we also notice that there's a lot of black dots on the outer nuclear membrane. And we wonder what those are. But now that we have 3D volumetric data, we can actually reconstruct it. And it's turned out that all those black dots that you see on the outer membrane of the nucleus is a pearl on string polyribosome structures sitting on the outer membrane. And you can also see the various nuclear pores there. And so that actually precipitated a very ambitious project at Janelia, which is to look at the cell interactome of all the organelles, and this is led by Jennifer Lippincott-Schwartz here. So we are one of the earliest image that they have collected is a HeLa cell. So basically we're milling through the entire HeLa cells and then basically annotate, and you can actually see the central here, another sister up here. So we can actually now annotate every single voxel in the system, in the image, so that you can account for every single biological structures. And here is a very, very small segment of the HeLa cell cytoplasm. And I'm going to play the other side. And you can also see the ER in lighter green and the ER exit site in the darker green color. You can see the cavity inside the ER network. And then here comes the microtubule. You can also see the holes inside the microtubules. And then here comes, I think, lysosomes of various smaller structures, endosomes, lysosome, and various vesicle. And as you can probably already predict, those bigger holes are reserved for the mitochondria. Okay. And so this we're milling more and more and more and more as we go along. Now, as some of you are looking at this, you're probably saying, wow, it would be really cool if we can do correlative light EM microscopy. Yes, we can. So here is again, the first time since Eric has won his Nobel Prize that Eric and Harold, good friends, come together and basically make this technique available. So the light microscopy part is basically structure illumination microscope for those who have taken the class. This is the microscopy that rely on the Fourier transformation, the interference pattern, but we're going to do it at cryo temperature. and so that the samples are immediately snap frozen so that we don't have to do chemical fixing. And then immediately after we have done all the super resolution at cryo temperature, we'll take it to focus ion beam and start milling. And so that is actually led to that cover on Science. So this is a mouse granule neurons. So this is actually a EM image, just pseudo color. And then here comes the super resolution image. And what you're going to see is basically drebin, which is a cytoskeleton membrane crosslinker, as well as the JAM, which is a cell-cell junction protein. What is interesting is as they mill in, they start to see electron dense and electron loosen area in the entire cytoplasm, in the membrane. We have no idea what it is, but when you pull it back, this is just membrane dense electron density at the junction of two cells, and we are still trying to figure out what the significance of these patches of different electron density is. So in a nutshell, these are the instruments that we have. Now I'm going to tell you how to get yourself to Janelia. Okay, so for those who don't know where Janelia is, we are very close to Washington, D.C. This is where the circus happened. And then this is the international airport of Washington, D.C., and Janelia is just right there, about 10 to 15 minutes away. And this is the Janelia campus. You can only see half of the research building because the other half is actually inside the hill. And this is the hotel, and this is the townhouses and studio. And if you go to AIC as a visitor, you will likely stay there. You have your own kitchen, you have your own washer, everything. So the program is attractive, and more importantly, I think beneficial to a lot of people, is because we will actually cover the cost of your experiment within reason. We will cover your lodging for 2 to 3 weeks. So the cost that you will have to incur yourself is your travel there, but you have to eat anyway, so it doesn't matter. So this is a good program. I always like to reassure everybody that those rooms really do have curtains. And so we are now entertaining a current call for proposal. To access the AIC, you have to write a proposal. It will be peer review. We have about twice a year of proposal call. Likely the next one will be announced sometime in November, and we'll give people about 2 months to write. And so likely at the end of January or so, that is the deadline, but likely that the proposal call will be announced sometime in November. The proposal is relatively easy to write. It's only about 1,000 words, 2 or 3 figures. It will go through a 2-tier review process. So the first tier will be just technical assessment by the AIC team. Some of them are standing behind there, so go sweet talk them if you want to. And we only assign a pass-fail grade, and so we ask 3 objective questions. Is can your questions be addressed by a commercial instrument? And if it is something that a standard confocal can address, then you kind of lose that justification to come to Janelia. Can the AIC instrument actually deliver the expected results? You can imagine people propose all sorts of pie-in-the-sky idea. Sometimes our instrument can deliver it. We can handle up to BSL-2, so you have to be careful to make sure that you don't bring Ebola in. And then if you pass, then we'll go to the second tier. That is when it will be reviewed more like a grant. We'll look at the scientific rationale, the experimental questions, whether or not it has the right justification for the use of the AIC instrument. Did you discuss the potential problems and alternative approaches? Again, you are there for 2 or 3 weeks. We don't have time to do a lot of troubleshooting. Quantitative approach. This is where most proposals come to die. You can imagine also that a lot of people just propose experiment to come to Janelia and generate eye candies after eye candies. We don't like that kind of proposal. So make sure that you have hypothesis-driven quantitative experiment, which every student at the workshop now know what to do. And then the amount of time, we don't really cap the amount of time, but it's usually 2 to 3 weeks. And personnel is important. Don't come alone. It's intense. Bring a couple of teammates because you will likely work 24/7. Because that instrument is yours 24/7. There is no competing users, and the application time, people like Chad, like Jesse, their time is completely dedicated to your project when you are there. So they don't entertain 2 groups simultaneously. more importantly, I think this is the last slide. More importantly, AIC is a lot more than just an imaging center. When you are there, you are considered a Janelia scientist. So everything that the Janelia has to offer institutionally, you get. Before you write a proposal, please talk to us. People who don't talk to us tend not to get their proposal accepted. The success rate is actually, if you don't talk to us and just randomly write a proposal, the failure rate is about 95%, so please don't do that. And then we will help you with a little bit of travel arrangement, especially visa waiver, and if we need you to write support letter, we'll do that. Before you arrive, sample will be shipped to Janelia already, right? So pre-arrival sample preparation like cell culture, even primary neurons, we will isolate it for you. Sample will usually be taken care of by Satya at the end. Lodging will be provided. Janelia also have all sorts of model organisms, and if you think that some of our lines are better, feel free to make a request. We have yet to turn down a single request. So sometimes people walk away with Drosophila lines, they walk away with zebrafish line, all sorts of stuff. Janelia has 54,000 Drosophila lines. Sometimes that might help. And also cDNA, we have the Michael Davidson library of about 5,000 fluorescently labeled cDNA, all sorts of model organism. Some of you are probably familiar with the Halo tag that Luke Lavis developed. The Halo tag that Janelia gave out is about 1,000 times more robust than what you get at Promega, and Promega charge you about $1,000 a tube. We give you a box. I always like to joke that you can use it till your graduate student retire. That is actually enough for you to it's a lot of dye, and it's actually cheap for us to synthesize it. Microscope time, as I mentioned before, is 24/7. You will be trained. You will actually be operating a lot of this advanced microscope after a day or two of training, but the support will be there for you, imaging assistance for sure. Even after you leave, the support continues. So actually, after you leave is where the struggle begins. You're going to be walking away, with tens upon tens of terabytes of data. So you need image analysis. Sometimes you need storage. Sometimes you need, compression. All that stuff will be able to help you. So I'm going to leave the last slide here with all the information. If you want to take a picture of that, but we would love to talk to you. We would love to have a lot more proposals coming from Latin America, and the applicant of the first successful Latin America proposal is sitting at the back. Marco, can you raise your hand? Hi. So, thank you. Question? Any question? Not just about science, about proposal, submission, anything. It all depends on what instrument you use. So some of the data are manageable. I wouldn't say small, I would say manageable. IPALM, sometimes people walk away with, in 2 or 3 weeks, about 10 terabytes, 6 or 7, 8 terabytes. The SynView light sheet, which is the mouse developing embryo, that instrument generates data at the rate of about terabyte per hour. So you're walking away with hundreds of terabytes of data. And unfortunately, that is the reality in the world. Everybody is preparing for it because you're not going to stop this technology from coming, and not just us, but EMBL and Max Planck. And as more and more biologists rely on this instrument because they are powerful. But to make it powerful, you have to have ways of handling those data. So data handling can come in two or three different facets, right? The very first one is data transfer. How are we going to transfer hundreds of terabytes? And we actually just ship the entire NAS tower to you. We have NAS tower of various sizes. I will just ship it to you with the appropriate size. You copy it to your home institution server, and then you ship the Nest Tower back to us. And so, if you are applying for instrument like the SynView, which is the mouse developing instrument, during technical consultation, we will ask whether or not you have that kit capacity at your home institution. Marco, for example, has also requested that for an extra week, I believe, to stay at Janelia, even after the imaging session is over, to be able to tap into the computing power of Janelia so that he can at least process and do some analysis before he comes home. So that is all totally acceptable. But it's coming. It's coming.
Good.
Yes.
Oh, thank you.
The microphone is just for recording. Yeah, it's not amplifying.
Okay. Okay, thank you very much for this. You show how it has been improving the temporal and spatial resolution. I was wondering now, what do you expect? I mean, is there a limit?
Is there a limit to its resolution?
You have
Yes.
to choose which would you try to make it better? Spatial, temporal resolution?
So the question is, we have discussed quite a bit about high spatial temporal resolution. I guess what you're asking is what is the next frontiers? The next frontiers come in two different fronts. I think the very first thing is the probe. At the end of the day, you will notice that almost every single breakthrough in microscopy not necessarily come from the optics side. It is actually the probes, the GFP and the blinking dye that makes all this thing available. So there are more and more dyes that biologists need. There is one very glaring omission, if you will, in probe development, which is biomechanical force probe. Nobody can see force very well. You can infer force from traction force assay, but to be able to really create a biosensor that is not Fred-based, please, that you can see force with ease. There are several people working on it. I'm hopeful that will be developed. That's number one. I think at some point we have to start using the available tools to start asking important questions. And I think image analysis have to keep up. We are starting to see more than what current software. If you see things that you cannot even describe in words, you cannot analyze it because you cannot articulate the idea. So you cannot, and so image analysis has got to keep up. If there is any kind of instrument development, and I think the focus should be and will be on how to keep your cell happy for as long as possible. Because biology don't happen on consistent rate. Cancer invasion, anything don't happen. It just a lot of nothingness, something happened excitingly, and then a lot of nothingness. And when that thing happened, it happened very, very quickly. And if you're trying to capture it really, really fast, the thing will die before something happened. If you capture it too slowly, when that happened, you may not see it. So the combination of being able to keep that sample happy for days, not just hours, and to have a smart microscope that will continuously survey at a very gentle level and until something happens, everything kicks in. I think that is the more likely, more useful scenario. Does that answer your question?
Thank you. Fantastic talk, like Marvel movies. So may I ask about this cell happiness and so on, whether you have some development tandems including atomic force microscopy?
Currently at Janelia, no, but it doesn't mean that it's not being developed elsewhere, right? And certainly, and Chad at the back, one of my team members is crazy about my biomechanical forces. So feel free to chat with him later. But currently, we don't really have a very good way to combine AFM with very high-speed imaging. And in fact, Chet was trying to develop one using sort of a lattice light sheet with a mirror to be able to combine that with the AFM. So,
Thank you.
right. Anybody else? Nobody asked me about proposal process? Yes. Ask away.
So you know, when you showed us these movies that are from development of Drosophila. This makes me recall swarm behavior.
Yes.
How do you analyze kind of behavior?
There are several
meaning-
ways. There are several ways. So, migratory behavior can be analyzed either at a single cell level, or as you say, probably as a cohort. So when you're analyzing cohort migration, then you will have to use a lot of cross-correlation and see how cells cross-correlate one another. So that is done quite a lot by a lot of people who are studying fluid dynamics, and so so that the algorithm and the knowledge is out there, but we have yet to fully take advantage of it and apply it in biology, especially in cell biology. Believe it or not, it is actually better applied in evolution and ecology, where people are studying birds and ants and stuff like that, than cell biology. But yes. Good.
Hi, absolutely fantastic. Thank you. Just perhaps a naive question. When you were showing I think it was a nerve of cord from the Drosophila, in which you showed the potato chips, the ones below seem to be more curly than the others above. Why is that? Because I was suspecting that
you may have missed, because it was sweeping very fast. Actually, the area where it got distorted the most is when light start entering from the outside in, because it's such a drastic change when it hit the sample. At that periphery, that's when it distorted the most. And also, the other thing, which is you have keen-eye, that's it, it's actually at the bottom, is also how the light sheet and everything is oriented. And that is sometimes at a very acute angle, that is when it has more chances of encountering all sorts of crap as it goes in. Yeah, so absolutely.
Okay, and then, may I have another one? When you mentioned about measuring the forces or seeing the forces, how I mean, in time, how close you can take pictures, for instance, I don't know, microtubules or whatever, in which can let you to guess that there is a motion and whether it's related with protein diffusion or something else.
So lattice light sheet can usually capture a volume, reasonable volume like a cell, and easily in about a second or so. So it's fast enough that you'll be able to see it. We have, there is an instrument that has been decommissioned that we no longer have, and that is actually heartbreaking story to decommission the original SIM system developed by Mats Gustafsson, but somebody has got to do it. We actually use this system to look at the neutrophil force as it is engulfing something, but not the lateral force, but the lift-up force. So that microscope has enough speed and gentleness, as well as the Z resolution, to see lift up the beats just a little bit. And so we can quantify that force. Yes, absolutely. Good. Nicholas?
I'm wondering whether you are thinking to generate some library of data about really, really important models, if you have identified that models. I'm thinking about some place where all of us can go and make a specific question with the same data, more or less like the model that people who do description of the universe works. You know, like
for the EM data, absolutely. Simply because EM data is not as specific as the fluorescent data. Fluorescent data, I'm not saying we shouldn't do it. I'm saying that fluorescent data tends to be very specific. Somebody generated a probe that has a mutation in it for a very specific isoform of something. And then outside of that is complete darkness, so you can't see it. But EM is a full context data that has a lot of opportunity and potential for people to mine, right? And so all this interactome data will be made public. In fact, the first one, the HeLa cells, is completely public. It's published in Nature. It's completely public. And we're going to continue to share it. And one of the things that we also hope that when people come to the AIC is that we will give you maybe a year or so that you have exclusive right to your data. And if that is a very important chunk of tissue, that maybe a year or two later, we will also share it as well.
All the synapses from a whole brain will be very, very interesting.
Yes. The synapses is actually already available. The entire Drosophila brain and big chunk of mouse brain is completely available, has been shared for free. Thank you.
Yeah. Very short. Regarding proposals, you get for in a poll, do you usually give any kind of feedback?
Absolutely. So this is actually a very important question, and I think it is important that I don't want people to feel intimidated by this process. The process, as Marco can tell you, it is about as simple as it gets. You just have to send us a very simple email. Hey, I'm interested in using your instrument. I don't know which one. That starts the process. And within 24 hours, one of us will contact you, and then we will sit down with you through Zoom. Sometimes the technical consultation can last an hour or two. We ask a lot of details, and then we help you formulate questions, just like I did with just half an hour ago, and to brainstorm together how to make the proposal successful. I mean, we cannot guarantee success, but we'll make it as successful as we possibly can. The number of proposal is very difficult to predict because depending on how many places I go, I'm pretty sure there's going to be a lot of proposal coming from Latin America. But I can tell you what the success rate is. The overall success rate is about 45%. So it's actually very good, precisely because people spend time talking to us. But like I said, if you don't talk to us, then almost all that fail are the one who never talk to us. So that is important. And we can entertain about 25, 30 projects per round, so about 50 to 60 projects a year.
Well, thank you very much, Leon.